Welcome to this explainer. Let's jump right in. Today, we're taking a truly fascinating journey that spans from the incredibly small all the way up to the staggeringly complex. We're going to bridge the gap from the microscopic, random drift of our DNA all the way up to macroscopic artificial intelligence models that can actually simulate living tissues. It's a massive story spanning biology, physics, and literally cutting-edge computation.
Let's kick things off with a pretty provocative question. Is most of your DNA actually just useless junk? Now, I know we often assume, kind of intuitively, that every single piece of our genetic code has been perfectly optimized by evolution, right? Like it all serves a specific survival-enhancing function. But what if that assumption is fundamentally flawed? What if the actual engine of biology is way messier than we like to admit?
Okay, let's dive into this. Here is our cognitive map for today's explainer. 1. Beyond Darwin, neutral theory. 2. The physics of soft tissues. 3. Graph networks in biology. 4. Decoding cellular communication networks. And finally, number 5, the future of biosimulation.
Starting with Section 1, Beyond Darwin, Neutral Theory.
To really understand the future of biological simulation, we actually have to step back in time, back to the 1960s, to a totally revolutionary concept by a scientist named Moto Kimura. See, for a long time, the dominant perspective in molecular biology was the adaptationist view. Think of it as a super tight feedback loop of natural selection and function. You have a trait, it provides an advantage, boom, it gets selected for. Simple, right? Well, Kimura's neutral theory completely flipped that script. He argued that down at the molecular level, stochasticity, just pure randomness, actually rules the game. Genetic drift and neutral mutations absolutely dominate the landscape. Basically, when selection is weak, random drift just takes over. And this means a massive portion of our genome might literally just be neutral, non-functional junk that drifted into place without giving us any survival advantage whatsoever. Evolution isn't just about the survival of the fittest. Down at the molecular level, it's actually heavily influenced by the survival of the luckiest.
Let's move to and see how this builds in Section 2, the physics of soft tissues.
Okay, so we are zooming out from the random drift of invisible molecules to the actual mechanical rules governing physical, biological forms. If we want to understand how our tissues hold their shape and resist stress, we absolutely have to look at this incredible concept called tensegrity. Imagine a structural principle that uses isolated components, like rigid struts under compression, and suspends them inside a continuous net of tension, kind of like elastic strings. It's like a giant flexible puzzle. Now, when researchers try to model simple epithelial tissues, like your skin, traditional continuous models often just fail under high strain. But by using these hexagonal tensegrity structures to mimic the cell cytoskeleton and its junctions, we can perfectly model that stress and strain. Our tissues, they aren't just solid blocks. They are incredibly dynamic, beautifully balanced mechanical networks.
And this brings us right to the science of multiphysics, which is essentially the bridge between biology and structural engineering. To build or simulate these shape-shifting soft materials, engineers use very specific external stimuli to trigger changes. For example, we can leverage the swelling of fluid-filled gels or trigger the electrical deformation of dielectric elastomers. We can carefully transmit mechanical stresses or even use morphing bilayer structures. By applying these specific physical forces, we can literally command materials to morph and fold exactly when and where we want them to. We are basically replicating the precise physical morphing we see in natural biological development.
Moving right along to Section 3, Graph Networks in Biology.
Now, we transition from physical structures to computational ones, stepping into the wild realm of hypergraphs. How do we track and simulate something as mind-bogglingly complex as the neural development of an embryo? Well, we build an emergent neural connectome, and we do it in 3 highly structured, action-oriented steps. Step 1, we format the raw biological data and filter out all the noise. Step 2, we construct spatial and temporal graphs. This is where we map out where the cells physically are and their lineage over time, literally connecting mother and daughter cells. And step 3, we apply graph attention networks. This takes all that messy, sparse data and converts it into dense embeddings. This allows the AI to learn exactly how and why those neural connections form over time.
And this brilliantly illustrates a core philosophy in modern biosimulation. Architectural elegance often defeats brute force complexity. You see, traditional AI models, they get really bogged down analyzing massive biological networks. Why? Because they rely on heavy positional encodings and these crazy complex message passing schemes that require just massive computation. But there's a newer model called edge set attention, or ESA, that essentially throws all of that away. It simplifies graph AI by focusing purely on the edges, the connections between the nodes. By dropping all that complex baggage, it actually outperforms the heavily fine-tuned complex models. It gives us a much clearer, far more accurate picture of the biology.
Section 4. Decoding cellular communication networks. So we're zooming out once again, this time to the macro level of how individual cells actually talk to each other. Now, what's really interesting about this slide is how we can systematically break this down across 5 cutting-edge spatiotemporal AI models that are literally reshaping the field right now. First, we have SGEL, which focuses on how dynamic brain networks evolve from random to highly structured. Then there's ESA, which we just covered, relying on the elegant simplicity of edge attention. Next up, self-referential GHNs. This introduces the wild concept of AI mutating its own code. Then we have CellSTN, which is an absolute powerhouse for spatiotemporal cell tracking. And finally, CellNest, which actually decodes the complex GSoC, the chemical relay networks happening between communicating cells. The big takeaway here is that these AIs, they don't just treat biology like a frozen snapshot, they treat it as a living, breathing, constantly changing system.
Let's look a bit closer at CellSTN, which acts as the ultimate cell tracker. Now, standard computer vision totally fails in biology. Why? Because cells squish, they move around, they divide, and they die in super unpredictable ways. CellSTN solves this by using bidirectional LSTMs. That means it literally scans biological video frames both forward and backward in time. So at 0 hours, it establishes cell locations. Fast forward to hour 6, and it's accurately tracking complex cell divisions. By hour 12, it's marking cell deaths. It's simultaneously learning what the cells look like and how they move. It's basically acting as this automated, multitasking tracker that understands cellular behavior completely holistically.
Which brings us to our final part, Section 5, the future of biosimulation.
We've reached a really fascinating point. What actually happens when the artificial intelligence we use to study biological evolution begins to mimic that evolution itself? This is the astonishing concept of self-referential AI. We are talking about neural networks, specifically graph hypernetworks, that exhibit emergent control over their own evolvability and mutation rates. So, the crucial point is, instead of a human tweaking the parameters, the network looks at its own structure, generates mutated versions of itself, tests them in the environment, and keeps the winners. If the simulated environment is nice and stable, the AI reduces its mutation rate. But if the environment becomes chaotic or throws up novel challenges, the AI cranks its mutation rate way up to find new strategies. The network itself literally becomes the evolutionary optimizer.
Which leads us to a profound thought to close out this explainer. We started this whole journey looking at the random stochastic drift of so-called junk DNA. Then we moved through physical tensegrity structures, up to AI decoding cellular gossip, and finally arrived at self-mutating code. It absolutely forces us to ask, will the next generation of AI evolve itself exactly like the biological systems it's trying to simulate? Are our computer models ultimately destined to become subject to the exact same evolutionary forces as organic life? It's an incredible frontier to consider, and it completely changes how we think about the intersection of biology and technology. Thank you so much for joining me for this explainer, and keep exploring these amazing connections.